And it's my pleasure to introduce Dr. Elliot Meyerowitz. He's the George W. Beadle Professor of Biology at California Institute for Technology. Welcome.
We're good. Thank you very much. I want to start out, before I even talk about the science, just saying for less than a minute that since I'm talking to an animal audience and I work on plants, that there are pressing human reasons why we have to understand how plants grow. There are about 1 billion people in the world who are malnourished because in the areas where they grow, we can't grow enough food for them to eat. And it's the primary cause of childhood mortality in the world. More people die of malnutrition every year than of malaria, AIDS, and tuberculosis combined. And they're children, not old people, like the people who die, like me, who will die of cancer most likely. I want to show this one slide just so you can look at it. There's a political reason, too. If you track food prices with time, revolutions always start when the food prices are high. The Arab Spring started with food riots. Venezuelan revolution that's going on now started with food riots. The Sudan revolution of 3 weeks ago started with food riots. We have to learn how to grow this stuff. And we don't fundamentally understand how plants grow. And so we can't do it from a basic point of view. You might say, well, America grows plenty of food, and we do. And we grow the food that's in deficit in a lot of other parts of the world, the parts that can afford to import it. But our agricultural mechanisms are entirely unsustainable. For every calorie of food you get on a plate or that I get on a plate, we use 10 calories of petroleum energy. 25% of our energy goes into growing our plants, into agriculture, and that's the contribution of agriculture, which should be taking up carbon dioxide, is in fact putting it out in vast quantities because petroleum is used to reach the yields we have. So we really have a pressing need to understand what's going on here. Now, my lab isn't going to do this. We're just little guys with a few postdocs, but what our contribution to it is to understand how the shoot apical meristem works, which is the collection of stem cells that sits at the end of every branch and puts out the stem, the leaves, and the flowers, so that it forms in the embryo, and through the growth of the plant, it puts out leaves and then pours out cells behind it, and it sits up at the top of the stem at all times. And then on its sides, it puts out leaves and then flowers. And this is a shoot apical meristem, sped up about 3,000 times to make it look like a lively rather than the stately object that it is. And it's making flower primordia on its periphery. And then we can trace patterns of gene expression in those flowers as they grow. And of course, we have a whole array of computer mechanisms by which we can segment the images, measure the volumes, track the cell divisions, and all this sort of thing. So we have a pretty good understanding of what happens in terms of gene expression and cell division and all these other parameters during the growth of the shoot apical meristem. And the reason we're interested in finding this is because we want to solve problems in developmental biology. How do patterns form in the shoot apical meristem? Patterns of cell division, gene expression, hormone response, and how does morphogenesis occur in plants? How do we get shapes of cytoskeletal organization, flower shapes, the position of primordia of flowers or leaves around the meristem, the phylotactic pattern. And I'm going to give you some solutions to that and talk about shape of tissues and a little bit about cell division and a bit about this too. And in the end, there are two areas I want to cover. One is showing you that growth leads to shape change and shape feeds back on growth. So a solution to a problem of morphogenesis. And you'll see that this also impacts on cell division and may provide some of the answers to Dagmar's questions, at least in plant epithelia. And then I'll talk about mechanical regulation of morphogen transport. And so we heard from Art about diffusion and from Dagmar about Turing models where morphogens are diffusing. Well, the system I'm going to talk about, of course they diffuse, everything diffuses, but there's also active and regulated transport of the morphogen that feeds back on itself to create self-organization in a way that, as far as I know, has not yet been discovered in animals, but it will be. Let's start with growth leads to shape change and shape feeds back on growth. Now, there's one thing I have to tell you as background information, which is plant cells are solid. They have a cell wall around them, and inside there's incompressible liquid that's at high turgor pressure, maybe 10 atmospheres in a shoot apical meristem cell. A champagne bottle is at 3 atmospheres. And so they're really pumped up, and that pressure is resisted by a cell wall, which is viscoelastic and has some yield strength and a variety of properties we can't get to in a short talk. The turgor pressure is not fully resisted by cells that are inside the tissues of a plant, but it's over-resisted by the cells in the epithelium, in the last layer of cells, the epidermis that sits on the outside of the plant. And so the epidermis, each individual cell is under turgor pressure and its wall is under tension, but there's an additional tension on the whole tissue, like on a balloon skin that pulls it to expand, so it's under, the whole tissue's under tension, and the walls of the epidermis also resist that additional tension and keep the whole plant in equilibrium. So it's critical to know that the epidermis is under tension, and this is the original demonstration from the 19th century, where if you take a dandelion stem and you cut it up the middle with a razor blade, it curls up like that, because the cells inside expand because they're no longer constrained by the extra resistance of the walls of the epidermal cells. And sometimes the expansion is so great that the epidermis tears off. Okay, given that, we can treat the epidermis of a shoot apical meristem as a thin-walled pressure vessel, so that we can calculate from the principles established by Cauchy and Euler 200 years ago that the stress in a hemisphere, a sphere, is isotropic. It's the same in all directions. But if you have a different shape, a shape like a cylinder, the stress is twice as much in this direction as in this direction, which is why when a boiler explodes, which is the concerns they had when they calculated this, the gap is this way and not that the top blows off. And why when you reinforce a cylinder, like a fiberglass cylinder, you reinforce it in this direction. We can take a shape of a shoot apical meristem and use this well-established mechanics, and we can see there's isotropic stress around the top of the meristem. Around the cylindrical part where it's making the stem, there's twice as much stress in this direction. And where a flower primordium is appearing on the side of the shoot apical meristem, there is very strong tension across the saddle point and less tension, theoretically, even potentially compression, but I don't think so in our case, orthogonal to that. And it turns out that the microtubules in the epidermis exactly follow this pattern, so that the microtubules are aligned parallel to the principal maximal direction of stress when stress is anisotropic. When the stress is isotropic, there's no preferred direction of the tubules, and they change their direction all the time. And let me underline that the microtubules I'm talking about are cortical microtubules that are wound around each cell just underneath the plasma membrane. And they reinforce, we'll get back to how they reinforce and what they reinforce. So they're changing their direction all the time here. They're rather fixed here. They're entirely fixed and bundled here. And they're parallel to the principal maximal direction of stress. So we can read out the stress patterns, and if microtubules are following them, we have a meter for the direction of stress, in addition to the shape of the tissue from which we can calculate it. So here's what some of the microtubules look like. And this is not a movie, and you can see they're in all sorts of directions up here. But if you look down over here in one of these saddle points, you can, at least the people in front can see that they're strongly aligned parallel to the expected stress direction. We can show that this mechanism is stress determined. We have finite element method models. And if we blow a hole out of the center with a laser, its wall stops being reinforced by continued synthesis, so the wall weakens. Because the epidermis is under tension, the hole tends to enlarge. The stress pattern goes from isotropic to anisotropic and circumferential to the wound, and you can watch the microtubules realigning to that in 2 or 3 or 4 hours. So we can change the stress pattern, and the microtubules follow exactly as predicted. Here's another example where we're not injuring, we're just squeezing it in a vise, and the same thing happens. Now, why is this important to the plant? And the answer is that the microtubules sitting just under the plasma membrane serve as tracks for cellulose synthase complexes, which project cellulose from under the plasma membrane through the plasma membrane and into the cell wall, so that the direction in which the microtubule sits defines the directions of synthesis of cellulose fibrils. And the cellulose fibrils are the strong tension-resisting parts of the cell walls. The elastic modulus of cellulose is about half that of steel. It's very inelastic. The yield strength isn't anything like that of steel. It'll pop if you pull on it, but it's very stiff. And so that if the cellulose is aligned parallel to the microtubules, it means that the cell wall is anisotropic and that it's constantly being remodeled to resist changing patterns of stress, so that it resists the stress, and the stress goes back into equilibrium after a gap of time when the cellulose gets synthesized parallel to the stress directions. And those little tiny dots are the cellulose synthase complexes. The big ones are the Golgi vesicles bringing the cellulose synthase up to the surface of the cell, and they're moving in the Z direction. We can look to see if this is really true, and so we can do our stress calculations on our shoot apical meristem, and then take a meristem and look at the direction of the cellulose fibrils using FESEM, field emission scanning electron microscopy. And in a wall like that, the cellulose fibrils are all aligned exactly with the microtubules. Remember, the top surface is free, and so the side walls are highly stressed anisotropically, and the microtubules and the cellulose align quite nicely to that. If we look at the top and the isotropic part of the meristem, there's no preferred alignment of the microtubules. But if we look at one of these saddle point regions where we freeze fractured it and then looked at the cellulose, it's highly aligned on the top surface, just as predicted by the model. So the predictions of the stress model are met because microtubules align to anisotropic stress and cellulose is deposited parallel to the microtubules. What does this mean for the shape of the tissue? Well, if we depolymerize the microtubules, we lose the shape. And all these valleys between primordia and meristem and the shape, the dome shape of the meristem all disappear over time, which means that the shape of the tissue is being maintained by the cellulose microfibrils, which follow from microtubule organization. And we can make models of this so that here's a meristem growing where stress is creating anisotropy in the cells with an FEM model. And here, where the stress is not being applied, and we expect different forms of growth. And in fact, we can grow meristems in exactly these conditions. We can add microtubule depolymerizing agents, and we get that spherical growth. The cells don't divide anymore. Or we can get normal growth. And so that we can predict the shape of the tissue from the stress pattern based on this mechanism. And it's a feedback mechanism because the shape is always changing. So the stress pattern is always changing. The cellulose reinforcement is always changing. And so shape goes to different shape, goes to different shape. And it's always feeding back from the global tensor field of stress to individual cells that respond individually to essentially the Gaussian curvature of the surface. So they know how the surface is curved near them because of the stress pattern. They make their walls anisotropic, and this changes their expansion direction because they expand orthogonally to the cellulose fibers because the cellulose fibers are stiff. And we can see this sort of thing going in even more complicated patterns. This is flower development, which is one of the frontiers we're working on now, and those are the sepals first coming out. And we can trace and track all of this, and so that we can follow every cell in this process and then calculate the stress patterns, which we've only done now for the earliest stages, and apply oryzolin or cellulose synthesis inhibitors at different stages of development. And I don't have time to talk about those experiments, but we can see just when the stress matters for the later stress response by timed experiments in a tissue with a complex shape like this. And so again, we have our models, and if we apply the stress calculations, we get realistic shapes of flowers, and if we don't apply the stress calculations, we don't. So we begin to develop models by which we can test our hypotheses and go back in and change shapes, remove cells, and do all the things that we were doing in the more simply shaped shoot apical meristem. There's something, there it is, and that's what it actually looks like. The tissue. Now, one other thing to mention while we're talking about shape is that the pattern of cell division also depends on this same mechanism, because when the microtubules are wrapped around a cell in these cortical arrays, what happens before division is that this array collapses to what's called the pre-prophase band, and the pre-prophase band determines the position in which the next cell wall will be inserted. And so that the new wall should always be parallel to the microtubule array, which it is. If we poke a hole in a meristem like this and then look a day later, we can see that all the new cell divisions have occurred circumferential to the hole, which they would not have otherwise. And so we can control cell division and we can control cell expansion through a mechanism that records the shape of the tissue, which is changed both by division and expansion, and feeds back on future shape. So we can understand morphogenesis as a mechanical process that feeds through cytoskeletal elements. One thing I should mention about this cell division pattern is that it's been known for more than 100 years that plant cells divide in response to stress. Leopold Knie in Germany in the 1890s showed that if you squeeze plants, you can determine the future patterns of cell division. This is the explanation for that old observation, we think at least. I don't know if he would have agreed. So here's summary one, our first mechanism, which is stress is generated by tissue shape and an epidermis in tension. The mechanical stress controls the cytoskeleton and thus the cellulose direction in the walls. Cellulose controls the subsequent cell expansion, which in turn changes the stress pattern. So we have our first novel developmental mechanism here, which is that tissue shape is sensed locally by cells through the tensor field of stress, leading to future changes in tissue shape. This would have been familiar to Einstein, who Wheeler summarized Einstein's theory of gravitation by saying that mass determines the shape of space, and the shape of space determines the transit of mass, the movement of mass. And it's exactly the same thing. And indeed, if we include stress and strain, our meristem has to be described as a fourth-order tensor field, just like space-time in Einstein's gravity theory. Oy. A little bit about mechanism, and then I'll move on. I'll just say a couple of things that we know about how these microtubules know to align to the stress pattern. One is that it's local. We can look at oddly shaped cells like these types of cells that are found in leaves and look at the microtubule pattern, and it matches the stress pattern in a subcellular way. That is, there are these lobes and necks in these cells. That's von Mies stress, so there's more stress overall there, which isn't a stress, actually. Principal component of stress is in this direction from neck to neck. In the areas in the middle, the second component of stress is in green, and so where the green crosses the red, those are sort of isotropic, and the microtubules aren't organized. So the pattern is subcellular. It happens locally within cells. And in some domains, the shape of the cell can determine the stress pattern, though in these leaves, If we poke a hole in the leaf, like we do over here, the microtubule pattern realigns circumferentially to the hole. So we go from a domain where the stress pattern in the microtubule organization is predominantly determined by turgor pressure and the odd shape of the cells. But if we injure the tissue, we get a more global response where we get supracellular organization of the microtubules that are resulting from the epidermis being in tension, so that we can go from one domain of stress to the other. And you can imagine how that might help plants to respond to their environmental conditions. Now, a couple of things about mechanism. If we don't allow microtubules severing, the microtubules won't realign, so when they cross over each other, one gets cut by catenin, and if that doesn't happen, we don't get our realignment, so that's one essential part of the process. And severing goes up about tenfold when we put the cells under mechanical stress, so that's another stress response. They start to cut their microtubules up in ways that make them align. And secondly, we can eliminate the activity of some formins, which in plants bind microtubules, that some bind actin, some bind other structures. There are 21 formins in Arabidopsis, more complicated than in animals. And if we prevent their activity, we can prevent this microtubule alignment also. So we have some clues about mechanism, but not the time to discuss them in detail. Let's go on to the second part, mechanical regulation of morphogen transport, where we start with the diffusing morphogen, just as in the last two slides, but we add an additional component to it, which is regulated and feedback organized active transport of the morphogen on top of the diffusion pattern. And the reason we got into this is that we wanted to solve the old problem of phylotactic pattern. It's been known since the ancient Greeks that leaves and flowers and things like that often are arranged in spiral patterns around meristems. Well, meristems weren't found until the 1870s to find. Well, the 1770s, Wolff had his punctum vegetationalis. But the individual flowers come out successively at an angle of 120 or 130 or 140 degrees from each other, one after the other, to create these regular patterns that we see here in Arabidopsis and with which we're familiar in some flowers and in pine cones and things of that sort. You'll remember from school that it had something to do with Fibonacci patterns. Actually, no, it doesn't. You also might have learned that primordia come out every 137.5 degrees. Give me a break. There are only 20 cells around, so it's got to be plus or minus 18. But if you measure a lot of them, they do tend to converge on that, quite remarkably. You'll see why in a second. Now, what is it that causes these organs like flowers to come out in this spiral pattern? And the answer has been known in part since the 1930s when Robin and Mary Snow showed that high levels of the plant hormone auxin are precursors to the appearance of a new leaf or flower primordium. And they showed this by taking dabs of auxin in lanolin and dabbing them onto meristems and showing that they would get a new primordium often in the position where they had dabbed the auxin. So high concentration of auxin leads to a new primordium. Now auxin induces new leaves and flowers, as I just said. Its immediate effect on cells in the shoot are to weaken the cell wall so the cells start to expand, and that's because it has a very rapid, just a few minute transcriptional effect to make proteins that activate proton pumps that pump acid out into the cell wall. Proton burns an ATP and puts a proton from the cytoplasm into the cell wall. Acidification of the wall weakens it, and so the cells start to expand. It also causes changes in gene activity through receptors that are found in the nucleus that are ubiquitin ligase-based receptors that are well understood. Now, there's another aspect of auxin which is key to the mechanism I want to describe, which is that auxin, which is indole-3 acetic acid, it's a simple tryptophan-derived molecule, is trapped in cells. And the reason it's trapped is that it's a weak acid, indole acetic acid, and so that at the neutral pH, it has a pK of around 5, and so at a neutral pH, it's dissociated from its proton, and it's an ion. And that ion will not go through the plasma membrane. So auxin's trapped in the cells. If it does get through the plasma membrane, which it can do because there are proteins that sit in the plasma membrane and let it out, called efflux carriers, it gets into the cell wall, and because of those proton pumps, the cell wall is a much lower pH. The auxin reassociates with its proton, about 50% anyway, and then it can diffuse back into the cell of origin or into the next cell. And so there's a acid trapping mechanism that's been known for a long time and which has been calculated in lines of cells. Graham Mitchison calculated in the early 1980s, analytically solved all the differential equations for transport in a single line, where the auxin efflux carriers are at the bottom of each cell. And so auxin has a net flux downward because it can get out of this cell and go back into the cell of origin, but it can go back into the next cell too, go into the next cell, and then it can only get out of the next cell when it gets to the bottom, and that creates a net flux. So auxin has its own circulatory system, and if we knock out the transport, one of the members of the transport carrier family, which are called pinform for this reason, this is the original pinform 1 mutant, and if we knock out pin 1, we don't get any flower primordia forming on the stem, because the auxin is not getting to high concentration, which means that at least one of the things that's necessary to get auxin to the right places in the shoot apical meristem to make the phylotactic pattern is auxin transport through this pin 1 mechanism. So we can label pin 1, and there are two things to mention about these live images with pin 1 GFP labeled. Remember, it's a plasma membrane protein, and it's asymmetrically distributed around the sidewalls of each cell. Where it's strongest is where most auxin is going out, and where it's weaker is where less auxin is going out. So we can draw arrows on this about the direction of auxin flux in any piece of tissue at any time based on looking at the pattern of expression of pin 1. The second thing to mention about pin 1 is that its gene is auxin-induced, so that when there's more auxin, there's more export, and when there's less auxin, there's less export. And we can use that property to look at the brightness of the pin 1 in any individual cell and infer from that what the auxin concentration is in that cell. And it's very rapidly induced in minutes by auxin. And so we get a pretty good readout of auxin concentration and of the direction of auxin flow from just looking at our confocal microscope images. And the general rule that we get, which you can see here, is that auxin is always moving from a lower auxin cell to a higher auxin cell. So while auxin diffuses perfectly well in the cells and in the cell walls, it's also being directed from lower parts of the gradient to upper parts of the gradient. And it's the ATP that's getting burned by those proton ATPases that provides the energy for this. There's no thermodynamic issue here. So our observations tell us is that local high auxin concentration causes new primordia to form, and it gets high locally by transport, and we have to consider diffusion, of course. Auxin efflux carrier moves auxin, and its gene is auxin-induced, so the rate of transport from a cell depends on the auxin level in the cell, and the auxin efflux carrier is polarized in cells and points towards neighboring cells with the highest auxin concentration. Does this account for the phylotactic pattern? Well, we can translate this into the language the plants speak and that the audience speaks. Those are the three things I just said. That's what's happening in the wall, which is a little bit more complicated. And yeah, exactly. We get exact reproduction of the auxin flux patterns and the phylotactic pattern that we saw in the living plants. And so we have at least a sufficient explanation for the phylotactic pattern. This is biology, so there's more, of course. There's other feedbacks involved, but there's no time to talk about those. Now, how is it that a cell knows the auxin concentration of its neighbors? I already told you the receptor is in its own nucleus, but it's directing the pin 1 protein to the plasma membrane against the wall that's shared with its highest auxin neighbors. And the answer seems to be that the highest auxin neighbors are expanding more rapidly, and so they're creating more stress on the common wall. And when we model pin 1 being drawn to the plasma membrane adjacent to the most stressed wall after an ablation, we get a radial pattern of pin 1 that should form around ablated cells. And if you watch, that's exactly what happens. We blew those 3 cells away, and the pin 1 gradually moves out to there, and then to there, and then to there. exactly going to the plasma membrane adjacent to the most stressed wall. And so we have another mechanical property, and we can show with many experiments that this is mechanical. Here's one where we've relieved the turgor pressure by putting some non-metabolizable sugar in the solution, and the pin 1 doesn't go anywhere when there's no turgor pressure. That is when the epidermis is not under tension. We can put a little bead that contains cellulase and digest the cellulose, so it reduces the stiffness of the walls. And we can put it there, and a few hours later, we get a radial pattern of pin 1 pointing outwards because these cells have expanded more rapidly, just as in the ablation experiment, than the cells surrounding them. And then we can wash the bead away, and everything goes back to normal, except sometimes the phyllotaxic pattern has taken a jog over to a new place. As you might expect, a phyllotaxis was determined by mechanical stress working through this regulated transport of morphogen mechanism. And so here's our second mechanism that auxin causes cell expansion. The resulting mechanical stress controls morphogen transport. New auxin peak, and it was the phyllotaxic problem for which Turing defined the term morphogen. So the Drosophila guys will tell me auxin isn't a morphogen because a morphogen has to activate different genes at different levels, but that is not the original Turing definition. New auxin peak changes which cells are expanding, thereby changing the auxin flow again. So it's another novel developmental mechanism, regulated transport of a morphogen. Just a word or two about mechanism. There's another mechanical response of meristems, and if we poke them with a needle right up there, let's see what we have to do to get this to start. Right up there, a wave of cytoplasmic calcium goes through the entire meristem, and then we withdraw the needle and a weaker wave goes through. So all these cells are talking to each other through another mechanical response, just like in any animal tissue, same sort of calcium stuff. We understand this calcium wave reasonably well, but I haven't the time to go through the modeling of it. Why do I bring it up now? It's because if we prevent the calcium wave from forming, pin 1 doesn't realign. So the first way that pin 1 knows its stresses change is this calcium transient. Is it involved in the movement of the protein, which is through vesicles, to new places in the plasma membrane? No, because if we do an experiment where we do an ablation and then we treat with lanthanum chloride to stop the calcium transient, just a few minutes later, by a few hours later, the pin 1 response is entirely normal. So it's only the very initiating event that depends on calcium. And that's about all we know about the mechanism at this point. We have a lot more to learn about this. Now, you might recognize that meristems aren't normally being prodded by needles, and so the stress source can't be needles poking meristems to make the phyllotaxic pattern. And I'll just show you that there's quite a lot of spontaneous calcium activity. This is not even sped up very much in the shoot apical meristem, and there's also fluctuations, these waves of calcium go through the meristem at about a 25-minute interval permanently, due to mechanisms which we have no understanding. And so there's a big frontier here. We don't understand yet the relation of cell stress or expansion to this calcium, but one imagines that there is a relation there. And this is another way that the cells know that they're expanding and therefore changing the stress pattern in the meristem. So with that, I'll stop. Here are the people in my lab, and some of the people who've done a lot of this work are Ting did the calcium work, and An did a lot of the calcium modeling. Pauline has done a lot of the microtubule work. Yuan has done a lot of the microtubule work. And of course, we couldn't do any of this without a lot of collaborators. And half the people here, they're all long-term collaborators in my laboratory from all over the place. Half of them have PhDs in physics and half have PhDs in biology. And we couldn't do any of this without them. Thank you.
We have time to take questions right here in the middle. Microphone's coming your way.
Hi, so super cool the calcium modeling also, which I hadn't seen yet. So a question, have you ever thought of poking it with 2 needles at 2 different places, just a little bit one after the other, or at the same time to see if you have these if this is excitable media, right? So you have the activation needs, and there's a delay, right? Would you be able to see the waves hitting and annihilating each other? I don't know, have you thought about such Is it technically possible? I know it's a very small region, but
Yeah, I think it's technically possible. We'd have to buy another micromanipulator, which we can't afford, but I think.
but your model would
I think we
predict,
could
right?
do it.
That
No, we're using one of these $3,000 Narashigi things to poke
Yeah.
it in there, and it's got a pressure sensor on it that we also can't afford another one of, but someday. But yeah.
but the model would predict that, right? So it would be
Yes,
like
yeah,
a typical
it would
excitable
absolutely.
media, and you could do these type of.
Yeah, I think the direction we're going with it immediately is that we've taken the computational model, which includes calcium pumps, pumping the calcium back down to levels. There are experiments I didn't show that the calcium is actually coming from the wall and going into the cells,
Of
and
course,
we
yeah.
can show that.
Can you get a sustained spiral ever? Does
No.
that never? You
We
don't see?
haven't done that. I think we'd have to keep poking because it damps out.
No, no. I mean,
after a
once you
couple of
break
drinks.
that target wave and you're able to, yeah.
Oh.
Well, we can.
I don't know. Maybe. But what we're doing is just mutagenizing all the ion channels and all the calcium pumps to see which ones are involved, and then we can go back in and really
And the
manipulate
link
the
to
process.
pins is just fantastic. No, so thank you. Yeah.
Yeah, thanks. That's a good idea. If you have a micromanipulator we can borrow from Cardiff.
Any other questions? In back? There you go.
Thanks. That was a great talk. I was curious if you tested, like, if you overwater the plants or underwater them, and how that changes the stressing, and if that changes cell shape.
If you overwater them, nothing happens really because they're already they're already wet normally. Once there's free water around, then the turgor pressure doesn't go up anymore. If you underwater them, they'll wilt. But we haven't really looked at the shape change of the cells. I don't think they change number of neighbors or anything like that. I'm sure of that. They're not that plastic. But they lose a little bit of volume. But we haven't really looked at shape. We've just looked at volume to make sure we've relieved the turgor pressure as control for those experiments I showed.
And we have one last
Good point.
question down here in front in the white shirt. Anna Becker, what's coming to you?
Yeah? No, here's the mic.
Correctly that you said oxen is diffusing from low concentration to high concentration?
No, it diffuses from high concentration to low concentration, but it's being actively transported from low concentration to high concentration at the same time. So the model, which I could bring up again, has diffusion in it going in one direction and active transport going in the other direction, determined by auxin concentration patterns itself. So that gives a positive feedback on auxin levels in cells. When a cell's high in auxin, it accumulates more auxin from its neighbors, but remember, it also induces its PIN1 gene, and so it starts to export more auxin that its neighbors are pumping in. And it's that feedback that gives the dynamism to the phylotactic pattern.
Thank you.
Thank you very much.